Imagine standing at the edge of the ocean, at night. The sky above you is a deep, quiet blue-black, scattered with stars. And the water is glowing. With every gentle wave that rolls toward the shore, a soft blue light blooms and fades. It pulses. It shimmers. And then it dissolves back into the dark, like a breath let out slowly. You take a step forward, and where your foot meets the wet sand, another small burst of light appears, right there, beneath you, and quietly disappears. You are not dreaming. This is real. And tonight, we are going to explore it together. Welcome. If you are watching this to unwind, to rest, to let the quiet hours carry you somewhere soft and still, then you are in exactly the right place. There is nowhere you need to be right now. Nothing that needs your attention. The world outside this moment can wait. All that exists for now is light. Gentle, living, breathing light. The phenomenon you are about to explore has been observed by human beings for thousands of years. Long before science gave it a name, people stood at shorelines just as you might imagine yourself standing now, watching the water glow, and felt something they could not explain. A sense of wonder, a sense of stillness, a feeling that the world was stranger and more beautiful than ordinary daylight ever let on. Ancient sailors wrote of seas that glowed in the dark. They called it the burning of the deep. Some believed it was the work of spirits. Others thought the ocean itself was alive with fire, a cool, harmless fire that did not consume, but simply shone. They were closer to the truth than they knew. The ocean is alive, and parts of it, on certain nights, in certain places, do something that seems almost impossible. They make their own light. Let yourself settle now, if you haven't already. Find a comfortable position. Let your shoulders drop. Let your jaw unclench. Let your hands rest soft and still. You don't need to hold on to anything tonight. There is a phenomenon that sailors and oceanographers have long referred to as the Milky Sea. It sounds like something from a fairy tale. And in a way, it is. Imagine you are aboard a ship, far from land, somewhere in the vast darkness of the Indian Ocean or the waters around Southeast Asia. It is the middle of the night. The crew is quiet. The engines hum low. And then, slowly, you begin to notice something. The water around the ship is changing. The deep black of the ocean is softening, becoming pale, then lighter, then luminous. Within an hour, in every direction you look, the sea is glowing a faint, steady blue-white. Not flashing, not sparkling, just glowing. Softly, continuously, as if the ocean floor had swallowed the moon. Some accounts describe the glow stretching to the horizon in all directions. Thousands of square kilometers of ocean, all lit from within. The ship moving silently, through it like a shadow passing through candlelight. These milky seas have even been captured by satellites orbiting far above the Earth. Instruments sensitive enough to detect the faintest emissions of light have photographed these glowing patches from space. Vast, drifting regions of ocean, radiating a quiet luminescence that can be seen from hundreds of kilometers above. Sailors who have witnessed it describe the experience as deeply, almost inexplicably peaceful. There is something about light that does not burn, that does not demand anything of you, that simply exists in the dark, that quiets something in the human mind. Perhaps you feel it now, even just imagining it. The slow, enormous glow, the silent ship, the sky above and the sea below, both full of soft light. But you do not need to sail to the middle of the Indian Ocean to witness living light. Some of it comes to the shore. On warm nights, in certain bays and coastlines around the world, in Puerto Rico, in the Maldives, in parts of California and New Zealand and beyond, the waves themselves seem to carry fire. Not the hot, orange fire of a flame. Something cooler, something bluer, something that moves with the water and vanishes as quietly as it came. This is the work of an extraordinary group of tiny organisms called dinoflagellates. They are single-celled. They are microscopic. And yet, when they are disturbed, when a wave crashes, when a fish turns sharply, when a swimmer moves through the water, they flash. Each individual organism produces only a tiny spark. A single flash, lasting less than 1/10 of a second. But there are millions of them. Hundreds of millions. Sometimes billions, gathered in shallow coastal waters, drifting in the current like an invisible, luminous crowd. And when a wave moves through them, when the water stirs and shifts, and every one of those tiny organisms responds at once, the whole surface of the sea lights up in a rippling cascade of blue. People who have swum in these bioluminescent bays describe it as one of the most dreamlike experiences of their lives. Every movement of your hand leaves a trail of light behind it. Your body is outlined in blue fire. You turn in the water, and the glow follows you, and fades, and blooms again. It is like swimming through a sky of falling stars. Except the stars are all around you. And the stars are alive. Let your breathing slow a little more now, if it hasn't already. In through the nose, slowly. And out. Imagine that water. Imagine the warmth of it. The soft salt smell of the air. The sound of small waves. And all around you, below, above the surface, along your arms and hands, that quiet, cool, blue glow. You are weightless. You are held. The water is warm and dark and full of living light. But the sea is not the only place where nature illuminates the night. Come with me now, away from the water, into the forest. It is late. The canopy above is thick and dark. The ground is soft with old leaves and moss. And somewhere, deep among the roots and the fallen logs and the damp hollows between trees, something is glowing. A pale greenish light. Faint at first. So faint you might mistake it for moonlight falling through a gap in the branches. But then your eyes adjust, and you see it more clearly. A soft, steady glow, coming from the bark of a fallen tree. From the moss growing at its base. From a cluster of small fungi growing where the wood has begun to soften with age. People have called this foxfire. It is a beautiful name for a beautiful thing. For centuries, travelers in forests across the American South, across Europe, across Japan and Southeast Asia, reported stumbling across this eerie, gentle glow in the dark of the woods. Children collected glowing wood and used it as a lantern. Miners, in the days before electric lights, are said to have used foxfire to dimly illuminate tunnels where open flames were too dangerous. The glow comes from fungi, from the living threads of mycelium that run through decaying wood, slowly breaking it down, slowly returning it to the earth. For reasons that scientists are still working to fully understand, some species of fungi produce light as part of this process. A steady, quiet, greenish glow that asks for nothing and attracts no attention, but is there in the dark if you look. There is something deeply calming about it. A light that is patient. A light that simply persists through the night, in the quiet of the forest, with no audience and no purpose that we can easily name. Just glowing. Just being. As if the forest itself is breathing, very slowly, and the light is the faint exhale. And then there are the fireflies. If you have ever seen them, you know. If you have not, close your eyes for a moment and try to picture this. A summer evening. The air is warm and a little humid, still holding the day's heat. The sky has gone from gold to lavender to a deep, soft blue. The first stars are appearing. And then at the edge of the treeline, just above the long grass, a small light blinks on. And then another. And another. Not constant. Not steady. Just little pulses of soft yellow-green. On. Off. Drifting upward. Drifting sideways. Each one moving at its own slow pace, following its own quiet path through the evening air. Within minutes, the field is full of them. Hundreds of blinking lights, rising and falling, drifting like sparks from a fire that burned away hours ago and left only this. A warm, unhurried afterglow, scattered across the whole meadow, rising slowly toward the stars. People travel to see this. They sit quietly in fields and on porches and at the edges of forests, and they watch. And they are still. There is something about bioluminescence, in all its forms, that invites stillness. It does not demand to be seen. It does not call out. It simply exists, quietly, in places where the ordinary light has gone. And when you find it, when your eyes are just enough to see it, something in you goes quiet too. Something that was tense eases. Something that was busy pauses. Something that was loud goes soft. That is where we begin. Not with formulas or diagrams. Not with a lecture or a list of facts. But with this feeling, this ancient, wordless feeling that human beings have always had when they stand in the dark and discover that something, somewhere, is glowing. Wonder. And wonder, as it turns out, is one of the most restful states a human mind can enter. Not excitement, wonder. The quiet kind. The kind that makes you want to sit very still and watch, and not speak, and just let the light do what it does. That is what we will carry with us through this entire journey tonight. As we explore the science of bioluminescence, as we travel from the deep ocean to the forest floor, from the bodies of tiny single-celled organisms to the flashing signals of fireflies on a summer night, we will keep coming back to this. The light, the quiet, the feeling of watching something extraordinary without needing to do anything about it. Just watching, just breathing, just resting in the soft and living glow. So settle in. Let your body grow heavy against whatever is beneath you. Let the weight of the day lift away. The glowing sea is waiting. The foxfire is patient in its forest. The fireflies are rising, one by one, into the warm, dark air. And we have all the time in the world. Go deeper now. Leave the shoreline behind. Leave the warm shallows, the sparkling surf, the soft sand beneath your feet. We are going down. Slowly. Gently. The way a single bubble descends in reverse, no rises. The way light itself moves through water. Quietly, and then more quietly, and then in a hush so complete that the word silence doesn't quite cover it. We are going into the ocean. Not the ocean you can see from a beach. Not the bright, turquoise, sunlit ocean of postcards and summer afternoons. We are going into the deep ocean. The old ocean. The one that has existed in near total darkness for billions of years, long before anything on land ever drew breath. And what we're going to find there is light. For a moment, imagine the descent. The surface is above you now, a shimmering ceiling of broken silver, growing smaller and more distant with every second. The light from the sun filters down in long, softening shafts, blue first, and then blue-green, and then a color that has no good name, something between teal and indigo, cool and still, and very, very quiet. At 100 meters, the shafts of sunlight are faint. At 200 meters, they are little more than a memory. At 300 meters, you are in a permanent blue twilight, a world that has not seen full daylight since the water was young. And at 500 meters, the last of the sun's light disappears entirely. You are in the twilight zone. This is one of the least visited places on Earth. Deeper than most submarines venture for casual exploration. Colder than anything that feels comfortable to the human body. Under pressure so immense that it would flatten an unprotected human being in seconds. And yet, it is full of life. Extraordinary life. Strange and slow and luminous life. Life that has found ways not just to survive in total darkness, but to thrive in it, to hunt in it, to hide in it, to communicate in it, to raise young in it. And the way much of this life has learned to do all of those things is with light. Let your breathing slow a little now. In slowly. And out. Picture that blue-black water around you. Not frightening, still. Enormous and still. The pressure of it like a gentle, total weight. Like being held. In the twilight zone, that band of water between roughly 500 and 1,000 meters down, scientists have made one of the most remarkable discoveries in the history of biology. Up to 90% of all animals living there produce their own light. 90%. Think about what that means. If you were to stand, somehow, impossibly, in a single square meter of that deep water, and watch everything that swam or drifted past you, 9 out of every 10 creatures you saw would be capable of generating their own bioluminescence. Not reflecting light. Not borrowing it. Making it. From within their own bodies, through chemistry alone, these animals create light in the most absolute darkness on the planet. Fish. Sharks. Squids. Jellyfish. Crustaceans. Small shrimp-like creatures carrying their own tiny lanterns. Marine worms, drifting in slow spirals, tracing arcs of cold blue light. Sea stars, faintly luminous along their arms, moving across the seafloor like slow-moving constellations. Microscopic algae, glowing as they drift. And bacteria, ancient, patient, living inside other organisms, lending their light like a gift. The deep ocean is not a dark desert. It is a galaxy. A slow, cold, impossibly deep galaxy of living light. Most of the light down here is blue. Or blue-green. Not because living things cannot make other colors. Some can, and we will come to them. But because blue is the color that travels farthest through seawater. Red light is absorbed quickly, within just a few meters of the surface. Orange and yellow follow. Green persists a little longer. But blue, particularly the wavelengths between about 470 and 490 nanometers, can travel through water with extraordinary efficiency. The ocean, in a sense, has its own preferred language of light, and the creatures living in it have evolved to speak it. Imagine moving through that world. The darkness is not complete. It never quite is. It is punctuated, constantly, by brief and gentle flashes. A spark here, A trailing glow there. Something in the mid-distance pulsing softly, once, twice, and then going dark again. Some creatures glow in patterns. Spots arranged in precise rows along a body, like the rivets on a hull. Stripes that run the length of a fish from eye to tail, perfectly symmetrical. Shimmering fields that seem to roll across a surface in slow waves, like a flag moving in a barely there breeze. These patterns are not random. They are as distinctive as faces, as individual as fingerprints. Members of the same species can recognize each other in total darkness by the specific arrangement of their lights, the spacing, the color, the rhythm of the flashing. In the deep ocean, your pattern of light is who you are. The organs that produce this light have a name, photophores. It is a beautiful word for a beautiful thing. From the Greek, phos, meaning light, and phoros, meaning bearing or carrying. A photophore is, quite literally, a light bearer. These are specialized cells, or clusters of cells, embedded in the skin or flesh of deep-sea animals. Some are simple, little more than a layer of light-producing cells backed by a reflective surface, like a tiny mirror behind a tiny lamp. Others are extraordinarily complex, complete with a lens to focus the light, a shutter to control its timing, a layer of pigment to direct it in a particular direction. Some fish carry hundreds of photophores along their undersides, arranged in precise geometric patterns. Others have a single large one, positioned like a headlight. Some jellyfish have rings of them along the margins of their bells, so that when they pulse and contract in the water, they look less like animals and more like something dreamed. Built-in lanterns, carried everywhere, never needing a battery, never going dim unless the creature wills it. And the creatures do will it. This is what makes bioluminescence more than just a passive glow. It is active. It is controlled. It is, in a deep sense, intentional. Animals regulate their light by controlling the flow of oxygen to the chemical reaction that produces it. More oxygen, more light. Less oxygen, darkness. The on-off switch is chemical, and it is precise, and many animals use it with extraordinary sophistication. They can flash. A single, sharp burst of light, lasting a fraction of a second, used to startle a predator or signal a mate. They can pulse. A rhythmic, repeating pattern, steady as a heartbeat, used to communicate across the dark water. Or they can glow steadily. A continuous, unwavering light, held for minutes or hours, used for purposes that require patience and persistence. Each mode of light serves a different purpose. And the ocean, across billions of years of evolution, has found uses for all of them. Let your body settle a little deeper now. If there is any tension remaining in your shoulders, your hands, your face, let it soften. You are very deep now. Very still. The water around you is vast and quiet. And the lights, when they come, are small and distant and endlessly calm. Consider the anglerfish. You may have seen pictures of it. That strange, deep-sea creature with the enormous jaws and the single glowing lure dangling from its forehead like a fishing rod grown directly from its skull. The lure glows. It dangles in the water above the fish's open mouth, swaying slightly with the current. And in the darkness of the deep ocean, that small, steady glow is extraordinarily visible. To smaller fish and crustaceans, moving through the dark in search of food, a tiny light can mean only one thing: something edible. A bioluminescent organism, small and slow and catchable. They move toward it. They follow it. They come closer. And the anglerfish waits. It does not chase. It does not hurry. It simply holds its light in the darkness, patient as stone, and waits for the dark to bring its dinner to it. This is light as a lure, perhaps the most dramatic use of bioluminescence in the ocean. But it is far from the only one. Some deep-sea squid carry photophores on the tips of their tentacles, trailing glowing filaments behind them like living fishing lines. Some worms produce flashes designed to mimic the bioluminescent signals of small organisms that larger creatures find irresistible. The deep ocean is full of deceptions, and most of them are made of light. But light is not only for hunting. It is also, in the deep ocean, a tool of escape. Some creatures, when threatened, do not go dark. They go brighter. Certain species of squid, when approached by a predator, release a cloud of bioluminescent fluid into the water. Not ink, as a surface squid might, but light. A glowing, diffuse cloud that hangs in the water, pulsing faintly, drawing the predator's attention while the squid itself slips away into the dark. Imagine that. A decoy made of light. A ghost left behind in the water, glowing softly, while the real animal vanishes. Others use their bioluminescence as a direct alarm, flashing so brightly when grabbed by a predator that the flash itself attracts larger predators, turning the attacker into the attacked. A scream made of light. There are even animals that, when disturbed, coat their attacker in bioluminescent slime, leaving them glowing in the darkness, conspicuous and exposed, while the prey quietly disappears. The deep ocean has been running these experiments for hundreds of millions of years. And still, it surprises us. Perhaps the most elegant use of bioluminescence in the ocean, though, is one that is not aggressive at all. It is a kind of invisibility. It is called counter-illumination. Here is the principle. When a creature swims through the upper layers of the ocean during daylight, or in the faint moonlit waters near the surface at night, its body casts a shadow downward. To any predator looking up from below, that shadow is a silhouette, a dark shape against a lighter background, a target, unless the creature can match the light above it. Lanternfish, small, silver-scaled fish that make one of the largest daily migrations on Earth, rising to the surface at night and descending to the deep by day, carry rows of photophores along their undersides. These photophores produce light that precisely matches the faint blue light filtering down from the surface above them. When a predator looks up, it sees nothing. No silhouette, no shadow, just the diffuse, even glow of the water. The lanternfish has erased itself with light. The bobtail squid does something even more remarkable. It doesn't produce this light itself, at least not entirely. Instead, it keeps a colony of bioluminescent bacteria living in a special organ in its mantle, called the light organ. The bacteria glow. The squid controls how much of that light escapes through the organ by adjusting a kind of biological shutter. At night, near the surface, the squid fine-tunes its bacterial glow to match the moonlight filtering down from above. An animal, carrying living bacteria, using their light to erase its own shadow. In moonlit water, this partnership, the squid and its glowing tenants, begins when the squid is barely hatched. The bacteria colonize the light organ within hours of birth. And from that moment, the two live together for the rest of the squid's life, each depending on the other. The ocean runs on these arrangements, on cooperation, on symbiosis, on the extraordinary willingness of life to find a way. Even in the dark, especially in the dark, there is something deeply restful about contemplating the deep ocean. Not because it is comfortable, it is not. Not because it is familiar, it is perhaps the least familiar place on Earth. But because it is so completely outside ordinary experience that the mind, trying to hold it, eventually lets go. You cannot worry about tomorrow while truly imagining the midnight zone. You cannot rehearse conversations or replay regrets while picturing a lanternfish erasing itself in the moonlit water. The deep ocean is too vast, too strange, too quietly magnificent for ordinary thought to hold on to. It simply dissolves the everyday. And what remains, when the everyday dissolves, is something quieter, something older, a kind of restfulness that doesn't need to be achieved or earned. It is just there, underneath the noise, waiting. Like light in the deep water. Patient, steady, always there. So let yourself drift, now, in that imagined darkness. Somewhere below you, in a world that has never seen the sun, creatures are moving through the black water, carrying their own small lights. They are flashing and pulsing and glowing steadily. They are luring and hiding and erasing their own shadows in the moonlight. They are doing what they have always done, what life has always done, finding extraordinary ways to exist and even to shine in the most difficult conditions imaginable. And up here, in the warmth and the quiet of wherever you are resting tonight, you are connected to all of it. The same chemistry that lights an anglerfish's lure. The same principles that make the bobtail squid invisible. The same ancient ocean from which, in some sense, all life on this planet eventually emerged. You carry that history with you. It is in every cell of your body. And tonight, it is here to help you rest. We rise now, slowly, gently, the way the deep ocean gradually gives way to warmer, lighter water as you drift upward. The pressure eases. The temperature climbs, almost imperceptibly. The blackness softens, first to a deep blue, then to something closer to the color of a clear sky seen from underwater. And then far above, the silver ceiling of the surface. We pass through it, and we are back in the world of air and land, and living things that breathe what we breathe. But the light, the living light, did not stay behind in the water. It came with us. It is easy to think of bioluminescence as a deep-sea secret, something that belongs to the dark and the pressure and the cold, to creatures so alien in form and habit that they feel like inhabitants of another planet entirely. And the deep ocean is, without question, where bioluminescence reaches its most extraordinary concentration. We will not forget what we found down there. But the light did not begin in the ocean, and it did not stay there. Over hundreds of millions of years, the chemistry of living light evolved independently, again and again, in organisms that never saw the deep sea. It appeared on land, in forests, in meadows, in the long grass at the edge of still water on warm summer evenings. And if you have ever watched the sun go down and waited, quietly, for what comes after, you may already know this. Let yourself settle again now. Feel the ground beneath you. Solid, still warm. The air is soft. Somewhere nearby, though you cannot quite hear it, there are leaves moving very slightly in a barely there breeze. It is summer. It is almost dark. And just above the meadow grass, something blinks. Fireflies. Of all the creatures that carry living light, fireflies may be the ones that feel most personal, most human, most like something from the edge of memory. A summer evening, a childhood garden, a field seen through a car window on a long, quiet drive. There is something about them that belongs to the softer hours, to the space between day and night, to the particular quality of dusk in late summer, when the air is warm and full of the smell of dry grass and distant rain. And yet, for all their gentle familiarity, fireflies are doing something quietly extraordinary. They are talking. Each flash a firefly produces is not random. It is not decorative. It is a message, precisely timed, carrying specific information to a specific audience. Male fireflies, flying low over the grass, flash in a pattern unique to their species. The timing of each flash, how long it lasts, how many times it repeats, how many seconds pass between each pulse, is a kind of code, a luminous signature, a way of saying, to anyone in the dark who speaks the same language, I am here. I am this kind. I am ready. And female fireflies, perched quietly in the grass, or on low branches, watch. They do not flash randomly in return. They wait. They observe. And when they see a pattern they recognize, a flash sequence that matches their own species, that says what they need it to say, they respond. A single, precisely timed flash. A reply sent back through the dark. If you were to watch a field of fireflies with this knowledge in mind, what had seemed like a random and lovely scattering of sparks would resolve into something more intricate. A conversation happening at the speed of light, across dozens of meters of warm summer air. Hundreds of small exchanges, each one a question and an answer, each one a connection forming in the dark. A field of fireflies is not a spectacle. It is a community, and the timing matters deeply. Biologists have measured the flash intervals of different firefly species with remarkable precision. One North American species flashes once every 5.5 seconds, in a long, sweeping arc. Another produces a rapid double flash. 2 short pulses, close together, repeated every few seconds. Another flashes in a slow, single blink, descending in a shallow curve through the air. These intervals are so consistent, and so species-specific, that an expert can identify a firefly by its flash pattern alone, in the field, in the dark, from a distance, as reliably as a birdwatcher might identify a song. Each species has its own rhythm, its own tempo, its own quiet signature written in light across the evening air. There is something almost musical about it. A field of fireflies in full display is, in a very real sense, a kind of composition, multiple rhythms layering over one another, each one distinct, the whole thing shifting and shimmering as the night deepens, and the temperature drops, and the grass stills, and the stars come out above it all. Let yourself hear that, for a moment. That imagined music made of light. The slow pulse of one species, the quick double flash of another, all of it drifting through the warm dark air above a summer meadow. And let yourself grow heavy, and still, and quiet along with it. Not all bioluminescence on land is as dynamic as the firefly's courtship dance. Some of it is slower, older feeling, more like something that has simply always been there, in the dark and the damp, patient and unhurried, and completely indifferent to being watched. Glowworms belong to this quieter category. The name is a little misleading. Glowworms are not worms at all, but the larvae and females of certain species of beetle or fly, depending on the part of the world you are in. But the name suits them, somehow. There is something worm-like about the way they rest, motionless, in dark places, cave ceilings, forest floors, the undersides of mossy banks, and simply glow. In New Zealand, in the cave systems of Waitomo and other limestone landscapes, there is a species of glowworm whose larvae produce a steady blue-green light from a small organ near the tip of the abdomen. The larvae hang from the cave ceiling by silken threads, sometimes hundreds of them, sometimes thousands, packed so closely together that the ceiling of the cave becomes a sky. A sky of still, cold blue-green stars. Tourists come from all over the world to float in silence through these caves on small boats, looking up. The guides ask them not to speak, not to use cameras, not to make unnecessary noise. And the visitors comply. Willingly. Naturally. Because the cave asks it of you. The light asks it of you. There is something about that soft, continuous glow above your head, cool as moonlight, completely silent, utterly still, that makes the idea of disturbing it feel almost unthinkable. You just look, and breathe, and let the ceiling of stars be what it is. The forest has its own version of this stillness. Foxfire, which we glimpsed briefly in our opening moments together, deserves a longer visit now. On certain nights, in old forests where fallenwood lies soft and damp among the leaf litter, the fungi that inhabit that decaying would produce a light that asks nothing of you. It does not flash. It does not pulse. It simply glows, a faint, diffuse, greenish luminescence that spreads along the threads of mycelium, running through the wood like something the wood is dreaming. Different species of fungi glow in different ways. Some glow only from their fruiting bodies. They're mushrooms, while others illuminate the entire network of fungal threads beneath the soil and bark. Some glow most brightly in young, fresh growth. Others glow steadily throughout their lives, brightening slightly in the warmth of summer and dimming in the cold. Scientists are still working to understand exactly why fungi glow. Unlike fireflies, whose bioluminescence has a clear and well-studied purpose, fungi seem to produce light almost incidentally, as a byproduct, perhaps, of certain chemical reactions happening within them. But there are hypotheses worth lingering on. One of them is beautiful. Some researchers believe that glowing fungi may use their bioluminescence to attract insects. Not to eat the insects. Fungi do not eat insects. But to use them. Certain insects, drawn to the glow on a dark forest night, land on the fruiting body or brush against the mycelium and depart carrying fungal spores with them. The glow, in this reading, is an invitation. A quiet advertisement in the dark. Come here. Take something with you when you go. The forest, it turns out, is as full of subtle communication as the deep ocean. It simply speaks more slowly, more gently, in a language of spores and roots and patient, persistent light. There is something worth noticing here, as we move from sea to shore to forest. Bioluminescence, wherever it appears, tends to belong to the patient things. The deep-sea creatures that glow steadily for hours, waiting. The firefly that repeats its signal, slow pulse by slow pulse, all evening long. The glowworm that hangs from a cave ceiling for months, softly shining. The fungus that glows through whole nights and seasons, asking nothing. These are not urgent creatures. They are not rushing. They have found that in the dark, patience is its own kind of power. That a steady, quiet light, held long enough, will find its audience. Will do its work. Will carry its message across the dark to wherever it needs to go. There is something in that worth carrying into sleep. The idea that not everything requires effort. That some things work best when they are simply held, steadily, without force or urgency. That a quiet light, patient in the dark, accomplishes more than you might expect. Your body knows how to rest. Your mind knows how to slow. You do not have to make sleep happen. You only have to let it. Like the fungi that glows without trying to glow. Like the glowworm that hangs in its cave and simply is. Like the firefly that blinks its one small signal into the summer dark, and then waits, without anxiety, for what the dark will bring back. Rest now a little deeper. Outside, wherever outside is for you tonight, the world has gone quiet. If it is summer, somewhere not too far away, there may be fireflies, lifting and blinking above grass that is cool with the night, having their slow, luminous conversations while the rest of the world sleeps. If it is another season, the foxfire is patient in its forest. The glowworms are hanging in their caves. The fungi are threading their quiet light through old wood in the dark of a forest floor that no one is watching tonight. All of this is happening, right now, whether anyone sees it or not. The living light does not require an audience. It simply shines. And that, in the end, is enough. By now, you have seen a great deal of light. You have stood at the edge of a glowing sea, and watched waves bloom blue in the dark. You have drifted through the deep ocean, past creatures that carry their own lanterns in the blackness. You have sat in a summer meadow and watched fireflies write their slow messages in the air. You have looked up at the ceiling of a cave and seen 10,000 steady green stars. And perhaps, somewhere in the back of your mind, a quiet question has been forming. Not an urgent question. Not the kind that demands an immediate answer. Just a gentle curiosity, turning slowly, the way a leaf turns in still water. How? How does a living creature make light? Not reflect it. Not borrow it from the sun, or the moon, or a fire somewhere nearby. But actually truly make it, from within its own body, from chemistry alone? Tonight, we are going to answer that question. Slowly, gently, without losing any of the wonder that brought us here. Because the answer, as it turns out, is every bit as beautiful as the light itself. Find a comfortable stillness, if you can. Let your eyes grow soft. Let the weight of your body settle completely into whatever is beneath you. We are not going anywhere dramatic now. We are going somewhere very small. We are going inside. Inside a single cell. Inside the quiet, warm, chemical darkness of a living body, where something extraordinary is about to happen. Light in the ordinary world comes from heat. A candle flame is hot. A light bulb filament is hot, so hot that the wire inside it glows. The sun is incomprehensibly hot. Even the screen you may have watched today, at some level, involves heat and electricity being converted into photons into light. Heat and light, in our everyday experience, are deeply connected, almost inseparable. But there is another way to make light. A cooler way, a quieter way, a way that does not burn, does not consume, does not require anything to be hot at all. It is called chemiluminescence. Light produced directly by a chemical reaction, without heat as an intermediary. You may have seen a simple version of this. Glow sticks, the ones sold at fairs and concerts, that you bend and shake and suddenly find glowing in your hand, work on this principle. Inside the plastic tube, two chemicals are kept separate until you bend it. The bending breaks an inner container, the chemicals mix, a reaction occurs, and light is produced. No heat, no flame, just chemistry, releasing its energy directly as light. Bioluminescence is chemiluminescence. But instead of two chemicals in a plastic tube, the reaction happens inside a living cell. And instead of lasting a few hours and going dark forever, it can happen again and again and again, thousands of times, in the same organism, on the same night, controlled with extraordinary precision. This is the quiet chemistry. And at its heart are two remarkable molecules. The first is called luciferin. The name comes from the Latin, lucifer, meaning light-bearer. The same root that gives us the word lucid, meaning clear and bright. Luciferin is the fuel, the raw material, the molecule that, when the conditions are right, will give up its energy as light. The second is called luciferase. Luciferase is an enzyme, a biological catalyst, a molecule whose job is to make a reaction happen more easily and more efficiently than it otherwise would. Luciferase takes luciferin and, in the presence of oxygen, facilitates the reaction that releases light. Together, they are an extraordinarily elegant system. Luciferin carries the energy. Luciferase enables the release. Oxygen is the key that opens the door. And what comes out the other side, instead of heat, instead of sound, instead of any of the other forms energy might take, is a photon. A single, tiny, perfect particle of light. What makes this particularly beautiful is how efficient it is. In most processes that produce light, a great deal of energy is wasted as heat. A traditional incandescent light bulb converts only about 5% of its electrical energy into visible light. The rest, 95%, becomes heat, radiating uselessly into the air. Bioluminescence wastes almost nothing. The chemical reaction inside a living cell converts most of its energy directly into light, with almost no heat produced at all. This is why the glow of a firefly does not warm your hand. Why the blue light of a bioluminescent wave does not feel like anything against your skin. Why you can cup a glowworm gently in your palm and feel nothing but the faint weight of it. Scientists call it cold light. Cold light. Light without fire. Light without cost. Light that gives everything it has and holds nothing back and still, leaves the creature that made it unharmed and ready to make more. There is something deeply peaceful in that image. A form of giving that does not deplete. A form of shining that does not burn. Let that idea rest with you for a moment. Cold light. Efficient, gentle, complete. A photon released without waste, without heat, without anything lost that needed to be there. Your breath is slow now. Your body is heavy and still. In some bioluminescent systems, particularly in fireflies, there is a third player in the reaction, beyond luciferin and luciferase. It is a molecule called ATP. ATP stands for adenosine triphosphate, and if you remember anything from a biology class you may have encountered it before. It is sometimes called the energy currency of the cell. Every living cell produces ATP, and uses it to power the work it needs to do. Moving, growing, repairing, signaling. In fireflies, ATP is required to activate the luciferin molecule before luciferase can do its work. The ATP essentially prepares the fuel for combustion, except that the combustion, of course, produces no heat. Just light. What this means is that the firefly's flash is not just a chemical reaction. It is powered by the same fundamental energy system that powers every other thing the firefly does. The same currency that moves its wings and drives its heart and processes its senses is also, in the right cells at the right moment, turned into light. The flash of a firefly is, in the most literal sense, a piece of the firefly's life force, converted, briefly and beautifully, into something you can see across a darkened meadow. And here is another quiet wonder. Luciferase, the enzyme that makes all of this possible, is not consumed by the reaction. Enzymes, by their nature, are catalysts. They enable a reaction without being permanently changed by it. When luciferase facilitates the conversion of luciferin and oxygen into light, the luciferase itself emerges from the reaction intact, unchanged, ready to do it again. A single molecule of luciferase can facilitate this reaction thousands of times before it finally wears out. Think about what that means for a firefly signaling on a summer evening. The enzymes in its light organ are working, over and over, each one enabling flash after flash after flash. The same molecules, doing the same quiet work, again and again through the whole long evening. There is something almost meditative about it. A repetition that is not mechanical, but alive. Each flash unique in its moment, produced by the same patient, tireless chemistry that produced the last one and will produce the next. Now, you might wonder, if bioluminescence is so elegant, so efficient, so useful, why haven't all living things evolved it? Part of the answer lies in the fact that luciferin is not one molecule. It is a family of them. Different organisms, having evolved bioluminescence independently of one another, have developed different chemical systems to produce their light. The luciferin used by a firefly is a different molecule from the luciferin used by a deep-sea fish. The system used by a glowing fungus is chemically distinct from the system used by a bioluminescent jellyfish. Two of the most widespread and well-studied luciferins have their own names. Firefly luciferin, the molecule used by fireflies and some other insects, produces that characteristic warm yellow-green glow. When it reacts with its corresponding luciferase and ATP, it emits light at a wavelength that sits right in the part of the visual spectrum most sensitive to the human eye. It is, in a sense, almost perfectly tuned to be seen. Coelenterosine, pronounced roughly as celientosine, is used by an enormous variety of marine organisms. Jellyfish, squid, certain crustaceans, many deep-sea fish. It tends to produce blue or blue-green light, which, as we have already explored, travels farthest through seawater. Some organisms that use coelenterosine do not even produce the molecule themselves. They obtain it by eating other organisms that do. The luciferin moves up through the food chain, passed from creature to creature like a quiet inheritance. Each system is its own solution to the same ancient problem: how to make light in the dark with nothing but chemistry. And life, as it always does, found not one answer but many. There is a particular quality to understanding something beautiful. It is different from simply seeing it. When you watch a firefly blink across a meadow without knowing anything about it, the experience is lovely, soft and nostalgic and warm. But when you know that the light comes from a molecule releasing a single photon as its energy shifts downward, that the enzyme enabling that release will do so again in a fraction of a second, that the whole thing is powered by the same ATP that moves the firefly's wings, the meadow doesn't become less magical. It becomes more so, because now the magic has texture. It has depth. It is not just a pretty light in the dark. It is a living chemistry, repeated 10,000 times in a single evening, in a single field, by creatures that have been doing exactly this for hundreds of millions of years. Understanding the mechanism does not close the door on wonder. It opens another one. Let your mind rest now among these ideas. Cold light, patient enzymes, molecules of luciferin waiting in living cells for the moment when oxygen arrives and the energy is finally, gently released. Somewhere tonight, right now, that chemistry is happening. In a forest, in a meadow, at the bottom of an ocean trench so deep that no sunlight has ever reached it. Quiet reactions, producing quiet light, in the quiet dark. The universe going about its ancient business. And you here, warm and still, a small part of the same extraordinary chemistry, breathing, resting, your own cells doing their own quiet work in the dark of your body. You are, in your own way, a chemical marvel. You are, in your own way, full of light. Rest now. Let the chemistry do what it does. There is a particular kind of peace that comes from thinking about deep time. Not the time measured in days or seasons or even lifetimes, but the other kind. The kind that moves in hundreds of millions of years. The kind in which entire mountain ranges rise and erode to nothing. In which continents drift like slow breathing. In which life, patient, relentless, endlessly inventive life, finds its way into every corner of the world and fills it with forms that no one could have predicted and no one could have designed. That is the time we are entering now. And the story we are following is the story of light. Not just how it works, but how it came to be. Why the universe, which began in darkness and silence, which ran for billions of years before anything alive existed in it, eventually produced creatures that could make their own glow. It is a long story, and it is a beautiful one, and we have all the time in the world to let it unfold. Settle in, just a little deeper. Let the weight of the world outside this moment drop away. We are going a long way back. By now, you know that bioluminescence has appeared in an extraordinary range of living things. Fish and fungi, fireflies and jellyfish, bacteria and squid and glowworms and sea stars, and 100 other forms of life, scattered across the tree of life like lights left on in a vast and darkened building. And a reasonable question, the kind of quiet, curious question that turns slowly in the mind without demanding an urgent answer, might be this. Did all of this light come from the same original source? Did one ancient ancestor, long ago, stumble upon the chemistry of living light and pass it down through the generations, branching and spreading across billions of years until it reached the firefly blinking in a summer meadow and the anglerfish waiting in the trench? The answer is no. And the answer being no is, in its own way, even more remarkable. Bioluminescence has evolved independently. Not once, not twice, not even a handful of times. Scientists studying the genetics and biochemistry of bioluminescent organisms have found that living light has arisen from scratch, from non-glowing ancestors, through entirely separate chemical pathways, at least 40 times in the history of life on Earth. Some estimates place the number considerably higher. 40 separate inventions of light. 40 different lineages of living things, in 40 different corners of the world, across hundreds of millions of years, each one arriving, through its own path, at the same extraordinary solution, the ability to make light from chemistry alone. This is what biologists call convergent evolution. The same outcome reached independently, by different routes. Like two rivers on opposite sides of a mountain that never meet, but both find their way to the sea. It happens throughout the natural world. Wings evolved independently in birds, bats, and insects. Eyes arose separately in vertebrates and mollusks. Warm-bloodedness appeared in both mammals and birds through different ancestral lines. Life keeps finding the same good ideas, over and over, because the universe keeps posing the same problems. But 40 times for bioluminescence is striking even by evolutionary standards. 40 times, life looked into the dark and decided, this would be better with light. The earliest of these inventions happened a very long time ago. How long, exactly, is difficult to say with certainty. The soft bodies of glowing organisms do not fossilize easily, and ancient bioluminescence leaves few traces in the geological record. But genetic analysis, the study of how distantly related organisms' DNA has changed over time, suggests that some bioluminescent systems arose hundreds of millions of years ago, possibly as long as 600 million years or more, deep in the Precambrian, before fish, before forests, before any animal had yet set foot on land. 600 million years. The Earth was a different place then. The continents wore different shapes. The oceans were different in their chemistry. The sky was different in its composition. And yet, in the waters of that ancient world, something was already glowing. Here is where the story takes a turn that I find endlessly wonderful. Scientists who study the origins of bioluminescence have proposed that the very first glowing organisms were not glowing for any of the reasons we might expect. Not to find a mate, not to hunt, not to hide, not to communicate, but to survive a poison. The poison was oxygen. In the earliest chapters of life on Earth, long before the atmosphere filled with the oxygen that we now breathe so easily, oxygen was not a resource. It was a threat. Early life had evolved in a world with very little free oxygen, and when photosynthetic organisms began releasing it as a byproduct of their chemistry, it was toxic to much of the life that existed at the time. Oxygen is, chemically, a highly reactive molecule. It can damage the delicate machinery of living cells. It can destroy proteins and membranes and the long, careful chains of DNA. The rise of oxygen in Earth's early atmosphere, an event sometimes called the Great Oxidation Event, was, for much of the life that then existed, a catastrophe. And so life, as it always does, adapted. Some organisms developed ways to neutralize oxygen before it could do damage. They evolved enzymes to capture and dismantle reactive oxygen molecules before they could wreak havoc in the cell. The hypothesis, and it is a hypothesis, but a well-supported and widely considered one, is that the very first luciferin and luciferase systems evolved as precisely this kind of protective mechanism. A way to grab oxygen and render it harmless through a controlled chemical reaction. The light, in this reading, was a side effect, a byproduct of the detoxification. Oxygen came in, the reaction occurred, and what came out was a molecule in an excited energy state, a molecule that, as it relaxed back to normal, released a photon, released light. Life was not trying to glow. Life was trying to survive. And in surviving, it found that it could shine. Let that idea breathe for a moment. The oldest light in living things, first kindled not as a signal, not as a lure, not as camouflage, but as an act of protection. A quiet chemical defense against a dangerous world. And from that accidental glow, over hundreds of millions of years, all the extraordinary uses of bioluminescence that we have explored together tonight eventually grew. There is something humbling about that. Something very still and very large. The firefly's flash. The anglerfish's lure. The bobtail squid's moonlit invisibility. The glowing cave ceiling in New Zealand. All of it, ultimately, descended from a single ancient act of survival. Life doing what it had to do to endure. And discovering, in the process, that it could be beautiful. Your breathing is slow now. Your body is heavy and warm. There is nowhere to go. Nothing to do. Only this long, quiet story to let wash over you. Once the chemistry of light existed, evolution, patient, tireless, working across timescales that dwarf anything we can easily imagine, found ways to use it. Hunting, hiding, communicating, defending. We have already met many examples of these strategies in our journey tonight. The anglerfish and its lure. The lanternfish erasing its silhouette. The firefly writing its signal in the summer air. The squid releasing its glowing decoy cloud. All of these are evolutionary solutions. Light being recruited, shaped, refined across millions of generations into something precise and purposeful. But there is one more category worth lingering on. One more way that life has found to use the chemistry of living light. Partnership. Not all organisms that glow make their own light. Some of them, like the bobtail squid we met in the deep ocean, rely on other living things to do it for them. Specifically, they rely on bacteria. Bioluminescent bacteria are among the oldest glowing organisms on Earth. They are found throughout the oceans, drifting freely in the water or living on the surface of dead and decaying matter, glowing steadily as they go about their ancient business. And some of these bacteria have entered into extraordinary arrangements with larger organisms. They live inside a host, in a specialized organ, a dedicated chamber within the host's body, kept warm and fed and protected. And in return they glow. The anglerfish's lure does not glow because the anglerfish makes light. It glows because the lure is colonized by bioluminescent bacteria, living inside a small bulb of tissue at the tip of the fishing rod appendage, producing their steady light in exchange for shelter and nutrition. The flashlight fish, a small, remarkable creature found in tropical and subtropical oceans, carries large light organs beneath each eye, so bright that the fish can be seen from considerable distances in the dark water. Those organs are not made of the fish's own light-producing cells. They are dense communities of glowing bacteria, tens of billions of them packed into two small pouches, glowing in symbiotic partnership with the fish that carries them. The flashlight fish can cover and uncover these organs with a fold of dark tissue, blinking them on and off as it needs to, using the bacteria's light as if it were its own, controlling when and how it shines. A tool made of living light, carried inside a living body. Two organisms so intertwined that it becomes difficult, in some meaningful sense, to say where one ends and the other begins. This is one of the things that bioluminescence keeps revealing, the deeper we look at it. Life is not a collection of separate, competing individuals. It is a web, an interconnection so vast and so ancient that the boundaries we instinctively draw between organisms, this creature and that one, this species and that species, begin to dissolve on close examination. The bobtail squid is not the bobtail squid without its bacteria. The flashlight fish does not shine without its glowing tenants. The deep ocean is not dark. It is lit collectively by millions of organisms, each contributing its small light to a shared darkness. And all of this, the web, the partnership, the ancient chemistry discovered in the act of surviving a poisonous atmosphere and refined over 600 million years into something of almost incomprehensible beauty. All of this is the story of bioluminescence. A story of life finding the same good idea, over and over. A story of light arising from darkness, again and again, by 1,000 different paths. A story of survival becoming beauty, and beauty becoming survival, across a span of time so deep that the mind can only hold it gently, loosely, the way you might hold water in your hands. Hold it that way now. Gently, without grasping. Let the deep time move through you like a slow current. 600 million years of living light, flowing through the dark, arriving here, at this moment, at your restful body, at the quiet behind your eyes. You are part of this story. Every living thing is. The same ancient chemistry that began as a defense against oxygen flows through the evolutionary history that connects you to every glowing creature we have visited tonight. You do not glow, not visibly, not like a firefly or a deep-sea fish, but you are made of the same patient, inventive, endlessly resourceful stuff that they are made of. Life, finding its way, shining when it can. Rest now in the deep time. Let the darkness be comfortable. Let it be, as it has always been for the glowing things of this world, not an absence, but a beginning. We have traveled a long way tonight. Down into the midnight ocean, past creatures that carry their own cold fire through the dark. Up through the water and into the warm air of a summer meadow, where fireflies were writing their slow signals in the dusk. Into the deep time of evolution, back to an ancient sea where the first living light appeared not as beauty, but as survival. And now, gently, the journey curves. Back toward us. Back toward human hands and human curiosity, and the remarkable things that happen when people look closely at something wonderful and ask, not just how does this work, but what else could it do? Because the story of bioluminescence does not belong only to the deep ocean and the forest floor. It belongs, in ways that are quiet and surprising and genuinely extraordinary, to us too. Let yourself rest in that thought for a moment. The same light that glows in the waves. The same chemistry that fires in a firefly's abdomen on a summer night. In laboratories. In hospitals. In the tools that scientists use, right now, tonight, while you are resting, to study the most intimate workings of living cells. The wonder and the science are not separate things. They never were. We have already met bioluminescent bacteria several times on our journey tonight. In the open ocean, drifting freely in vast, slowly glowing clouds that sailors once called the Milky Sea. Inside the light organs of the bobtail squid, carefully tended and precisely controlled. Packed in their billions behind the eyes of the flashlight fish, blinking on and off in the dark water. Bioluminescent bacteria are ancient. They are extraordinarily widespread. And they are, in a sense, the most democratic of all glowing organisms. They do not require a complex body or a specialized organ or millions of years of evolutionary refinement to produce their light. They simply glow, steadily, as part of their ordinary chemistry, as naturally as breathing. In the open ocean, free-living bioluminescent bacteria drift in the surface waters, contributing their faint, continuous glow to the broader luminescence of the sea. They colonize the surfaces of dead fish and floating organic matter, glowing as they feed and multiply, producing the eerie shimmer that sailors sometimes noticed around debris in the water. And they colonize living hosts, not just squid and fish, but a remarkable variety of marine organisms, each relationship shaped by millions of years of coevolution into something precise and mutual and quietly astonishing. The bacteria need a home, a stable, warm, nutrient-rich environment where they can live and reproduce without the uncertainties of the open ocean. The host needs light, for camouflage, for communication, for luring prey. And so the two find each other, recognize each other through chemical signals that have been tuned across evolutionary time, and enter into an arrangement that benefits them both. Neither one planned it. Neither one chose it, in any sense we would recognize as choice. It is simply what life does, given enough time. It finds the arrangements that work. It keeps them. It refines them. Until what began as a chance encounter between a drifting bacterium and a newly hatched squid becomes a partnership so deep and so necessary that neither can easily survive without the other. There is a kind of rest in contemplating these arrangements. The absence of urgency in them. The patience. The way they developed not through effort or intention, but through the slow, quiet accumulation of what worked. Generation after generation. Across timescales that swallow our ordinary sense of time entirely. Nothing was forced. Nothing was rushed. It simply became. The way sleep becomes, when you stop trying to make it happen, and simply allow the body to do what it already knows how to do. Now let us step quietly into a laboratory. Not a cold or clinical place. Not tonight. Tonight, a laboratory is simply a room where human curiosity has been given tools and time. Where people who find the natural world endlessly fascinating have gathered to look more closely, to ask better questions, to follow the light wherever it leads. And in the second half of the 20th century, the light led somewhere remarkable. Scientists studying bioluminescence had long understood the basic chemistry: luciferin, luciferase, oxygen, the efficient release of a photon. But as the tools of molecular biology became more powerful, researchers began to ask a new question. Not just how do living things make light, but what if we could use that chemistry ourselves? What if the light that evolution had spent hundreds of millions of years refining could become a tool in the hands of the people trying to understand life? It could. It can. It does. Luciferase, the enzyme at the heart of bioluminescent chemistry, has become one of the most widely used tools in modern biology and medicine. Here is the elegance of it. Scientists can take the gene that encodes luciferase, the biological instruction, written in DNA, that tells a cell how to build this enzyme, and attach it to another gene they want to study. Any gene. The gene that controls how a cancer cell divides. The gene that a virus uses to enter a cell. The gene that switches on during the development of an embryo. Any gene, anywhere in the genome of any organism. When that attached gene is active, when the cell is reading it, following its instructions, doing whatever that gene instructs it to do, the luciferase is produced alongside it. And wherever luciferase is produced, in the presence of its luciferin fuel, there is light. The cell glows. In the dark of a laboratory camera, a single glowing cell is visible. A cluster of cells, following a particular genetic instruction, lights up like a small constellation. A tumor expressing a particular protein announces itself with a faint, cold blue-green shine. Suddenly, processes that had been invisible, happening in the dark interior of cells, in the molecular machinery that no microscope alone could easily follow, become visible, trackable, measurable, understandable in ways that transform what medicine and biology can do. Luciferase reporters, as these tools are called, are used in thousands of laboratories worldwide. They have been used to study how infections spread through the body, how drugs interact with specific genes, how embryos develop from a single fertilized cell into complex organisms, how the brain's cells change with age or disease. They have accelerated the development of treatments for cancer, for viral infections, for genetic disorders. The light of a firefly, refined by evolution across 300 million years, now illuminating the interior of a human cancer cell in a research hospital. The wonder does not diminish. It deepens. But perhaps the most famous contribution of bioluminescence to human science is not luciferase at all. It is a protein discovered in a jellyfish. The jellyfish is called Equoria victoria, a small, unassuming creature found in the cool waters of the Pacific Ocean, translucent and drifting, unremarkable in appearance to any casual observer. For most of its existence, it was virtually unstudied. But Equoria victoria glows. And in the 1960s, a scientist named Osamu Shimomura, working at the Friday Harbor Laboratories in Washington state, became determined to understand why. What Shimomura found, after years of painstaking work that involved collecting and processing hundreds of thousands of individual jellyfish, were two things. The first was a protein called aquarin, which produced blue light in the presence of calcium. Interesting, useful, but not what changed the world. The second was something he almost overlooked. A second protein in the jellyfish, one that absorbed the blue light from aquarin and re-emitted it as a bright, vivid green. A protein that, unlike aquarin, did not need any chemical fuel to produce its glow. It simply needed light to come in, and it would produce light, green light, reliably, predictably, in return. He called it green fluorescent protein. GFP. For some years, GFP was a curiosity, a beautiful chemical footnote in the study of an obscure jellyfish. And then, in the 1990s, other scientists began to realize what it could do. Because GFP, unlike luciferase, does not require any additional chemicals to function. It is self-contained, stable, remarkably robust, and its gene, like the gene for luciferase, can be attached to any other gene in any other organism. When scientists attached the GFP gene to a gene in a bacterium, the bacterium glowed green wherever and whenever that gene was active. When they attached it to a gene in a developing embryo, they could watch, in real time, as individual cells switched that gene on and off during development, each one briefly glowing to announce its activity, like a tiny green lamp switched on in a dark room. When they attached it to a protein in a living neuron, they could watch, under a microscope, as that protein moved through the cell, along the long, branching arms of the neuron, through the gaps between cells at synapses, through the extraordinary interior geography of the brain. For the first time in history, scientists could watch the living cell do its work in real time. Not frozen in a snapshot, not inferred from chemical analysis after the fact, but alive, moving, glowing, moment by moment, in the light of a protein borrowed from a drifting jellyfish in the cold Pacific Ocean. In 2008, Osamu Shimomura and the two scientists who developed GFP as a biological tool, Martin Cholfi and Roger Chen, were awarded the Nobel Prize in Chemistry. The citation noted that GFP had become one of the most important tools in contemporary bioscience. A small, unremarkable jellyfish. A patient scientist collecting hundreds of thousands of them on a Washington coastline. And a protein that, without anyone planning it, without anyone knowing what it would eventually become, contained within itself the seeds of a revolution in how we understand life. This is what bioluminescence keeps doing. It keeps giving more than it appears to offer. A glowing wave that turns out to be millions of individual organisms, each one flashing in response to movement. A steady foxfire in the forest that turns out to be a network of fungal threads, threading light through decaying wood for reasons we are still working to understand. A jellyfish that turns out to be carrying a protein that will, decades later, illuminate the interior of a living brain. The light is never just light. It is always also information, always also connection, always also a door, left slightly open, into something deeper. Your eyes are heavy now. Your breathing is slow and even. The laboratory is quiet tonight. The jellyfish are drifting in their cold Pacific water. The fireflies have found their partners in the summer dark, and the meadow has grown still. And somewhere, in a warm room with gentle instruments and soft light, a scientist is watching a cell glow green. A single cell, in a dish, doing what cells do, illuminated by a protein that a jellyfish made, for reasons of its own, in an ocean that has been glowing long before any of us arrived to notice. We arrived. We noticed. We followed the light. And it led us here, deeper into the mystery of life than we had any right to expect to go. Deeper into wonder. Deeper into understanding. Deeper into the extraordinary, ongoing, endlessly surprising conversation between human curiosity and the natural world. Rest in that now. The conversation continues, whether you are awake to follow it or not. The world is working, quietly, in every direction. The light is doing what it does. And you are allowed, completely and without reservation, to simply rest. We have come a long way tonight. And yet, somehow, we have arrived back exactly where we started. At the edge of the water, in the quiet dark, watching something glow. Do you remember the beginning? The image we started with, before the science, before the chemistry, before the deep time and the ancient ocean and the jellyfish and its luminous secret, before all of it. Just the shore. Just the night. Just a wave rolling in slowly from the dark, and blooming, briefly, with a soft blue light. And the feeling that came with it. That particular stillness. That wordless sense that the world was stranger and more beautiful than ordinary daylight let on. That something had been here, glowing quietly, long before anyone arrived to see it. And would continue glowing, just as quietly, long after everyone had gone. We started there. And here we are again. Let your body be completely still now. Not the stillness of effort, not holding yourself still, but the other kind. The kind that arrives on its own when the mind finally lets go of the day. When the list of things and the weight of things and the texture of everything that happened and everything that didn't happen simply softens. And what remains is just this. Breathing. Warmth. The dark behind your eyes. And somewhere, in that dark, a faint and quiet glow. Right now, as you rest, the ocean is doing what it has always done. On the surface, In warm, shallow bays where the water is thick with microscopic life, the dinoflagellates are waiting. And somewhere tonight, on a coastline you may never visit, in water you may never touch, a wave is breaking. And in the breaking, 10 million tiny organisms are flashing at once. A brief, brilliant bloom of blue light that rises with the wave and dissolves with the foam, and is gone before anyone watching could quite say they saw it. And then another wave comes. And it happens again. It has been happening this way for hundreds of millions of years. The same chemistry, the same cold blue flash, the same patient in different beauty. Before there were eyes to see it, before there was a word for it, before anyone thought to wonder why. The ocean does not glow for us. It simply glows. Deeper down, in water that no sunlight has touched since the oceans were young, the creatures of the twilight zone are going about the ancient business of their lives. The lanternfish are rising, as they do every night, following the slow vertical migration that takes them from the deep cold toward the surface, drawn by the faint light of dusk, drawn by the thin soup of microscopic life that gathers in the upper layers after dark. Their photophores are lit, row after row of small, precise blue lights along their bellies, matching the faint moonlight filtering down from above, erasing their silhouettes, making them invisible to whatever might be looking up. They do not know they are beautiful. They do not know that anything is watching. They are simply doing what they do, as they have always done, in the dark and the pressure and the cold. Somewhere else in that deep water, a jellyfish is pulsing. Its bell contracts and expands with the slow, hypnotic rhythm of something that has no need to hurry. With each contraction, a faint ring of light moves around the margin of its bell, cool and blue and perfectly even, and then fades. Contract. Glow. Fade. Contract. Glow. Fade. Like breathing. Like a heartbeat. Like the rhythm of something so deeply alive that life itself seems almost to be the wrong word for it. It is older than that. It is something the water has been doing, in one form or another, for longer than we can hold in our minds. In the forest, the fungi are patient. The foxfire glows in the dark of a fallen log, threading its pale green light through wood that is slowly, gently, returning to earth. There is no one in the forest tonight. No one to see it. No eyes adjusted to the dark. No quiet footstep pausing on the path. The fungi glow anyway. The light does not need a witness. It does not require acknowledgement or appreciation, or even understanding. It simply is a quiet chemical fact, a small and ancient process, happening in the dark as it has always happened. And in the morning, when the sun comes up and the green light is swallowed by the ordinary brightness of the day, the fungi will still be there, still threading through the wood, still slowly, patiently doing their work. The glow invisible in the daylight, but present, always present, waiting for the dark to come back and make it visible again. In the meadow, the fireflies have quieted. The long conversations of the evening, all those careful signals, all those precise intervals, all that luminous courtship conducted in the warm air above the grass, have wound down with the cooling of the night. The males have settled. The females are still. The meadow is dark and damp and full of the smell of earth and late summer. But in a few hours, when the warmth returns with morning, the chemistry will still be there, waiting in small, patient bodies in the grass, ready to begin again when the time is right. The firefly does not worry about whether it will glow again. It simply carries the chemistry. It carries the light, ready and waiting, until the evening comes. Let your breath be slow. Let it be easy. In and out. In and out. This is what tonight has been about, really. Not just the science, though the science is extraordinary, and we have followed it from ancient oceans to modern laboratories and found wonder at every turn. Not just the beauty, though the beauty is real, and the images we have built together tonight are worth returning to, in any quiet moment, for as long as you want them. But this, the reminder that the dark is not empty, that even in the deepest, most lightless places on this planet, in the trenches of the ocean where the pressure would crush anything we built, in the hearts of old forests where no human has ever walked, life has found ways to carry its own illumination. Silent illumination, patient illumination, Illumination that asks nothing and demands nothing, and has been quietly present in the dark long before anyone arrived to name it. Bioluminescence is, in a very old and very deep sense, a story about resilience. About the discovery that darkness is not a problem to be solved. It is a condition to be adapted to. A circumstance that, given enough time and enough ingenuity, and enough of the extraordinary resourcefulness that seems to be woven into the fabric of life itself, can be met with light. Not loud light, not harsh light, not the light that overwhelms and bleaches and makes everything flat and ordinary, but the other kind, cold light, quiet light, the light that glows from within, the light that costs almost nothing and wastes nothing and simply, steadily, illuminates whatever is immediately around it. Not the whole world, not forever, just here, just now, just enough. You carry something like that too. Not literally. Not the luciferin, not the enzyme, not the cold blue flash in the dark water. But something in the same spirit. Something that, even on the difficult nights, even in the seasons when the darkness feels heavier than usual, is still there. Still patient. Still present. Still capable, when the conditions are right, of illuminating the space immediately around it. You do not have to glow at full brightness all the time. The firefly rests between flashes. The lanternfish descends back into the cold each morning. The foxfire is invisible in daylight. Bioluminescence, in almost every form we have encountered tonight, is intermittent. It glows when it needs to. It rests when it doesn't. And the resting is not a failure of the light. It is simply part of the rhythm. Rest is part of the rhythm. This rest, right now, is part of your rhythm. So let the ocean glow tonight without you watching. Let the deep-sea creatures follow their ancient migrations in the dark. Let the fireflies rest in the grass, their chemistry waiting, patient and ready. Let the foxfire thread its quiet light through the old wood in the forest. Let the jellyfish pulse in the cold black water, contracting and glowing and fading, over and over, in the dark. All of this will continue. It has always continued. The living light of this planet does not need your attention to persist. It has been glowing since long before you arrived, and it will be glowing still, long after tonight has passed into morning. You are allowed to let it go on, without you for a while. You are allowed to be still. You are allowed to rest. Somewhere, right now, a wave is breaking on a dark shore. And in the breaking, for just a moment, brief and quiet and completely without effort, the water glows. Blue. And then gone. And then the next wave comes. And it glows again. This is how it has always been. This is how it will always be. The light, and the dark, and the light again. Patient and ancient and endlessly, quietly, beautifully alive. Good night.